This video is intended to provide a brief general introduction to the basic principles of the structure and properties of organic compounds. Now, why all this focus on chemistry? Last week we spent a lot of time reviewing general chemistry. Now we're turning our attention to organic chemistry. Well, very simply, it's because if you break it down to the most fundamental level, everything that happens in biology is chemistry. All of those interactions and reactions that are happening at the molecular level are the basis for all of the processes of life occurring at the cellular or organismal level. So if we want to understand those functions that are occurring in living organisms, we have to investigate the structure and properties of the organic compounds, those carbon-based compounds that living organisms are made of. Now, it's no accident that biological molecules are based on a carbon skeleton. In fact, given carbon's atomic structure and properties, it makes a lot of sense that that would be the case. You'll recall that carbon's atomic number is 6, which is based on the number of protons in the nucleus, of course, and the most common isotope of carbon also has 6 neutrons in the nucleus. So if we look at a carbon atom nucleus, we'd expect to see 6 positively charged protons and 6 electrically neutral neutrons. We also know that an atom in an unreacted state has a number of electrons outside the atomic nucleus that's equal to the number of protons in the nucleus. So in that carbon atom, we'd see 6 electrons, 2 in the innermost electron shell, or first shell, and the remaining 4 in the second shell, which is the valence shell for carbon. Valence shell, as you'll recall, is the outermost electron shell that contains electrons. And as we've talked about before, an unfilled valence leads to reactivity in an element. Because of carbon's relative electronegativity compared to its most common partners, oxygen, nitrogen, and hydrogen, the types of reactions we see with carbon are covalent bonding interactions with up to 4 atomic partners to share 4 electrons, achieving a completed octet. And as we know, a filled valence, a completed octet or duet, leads to stability. So carbon's tetravalence leads to the capacity to form 4 bonds, which creates a potential for large and structurally diverse molecules. The fact that the electronegativity difference between carbon and its most common biological atomic partners is relatively small leads to stable covalent bonds and therefore stable biomolecules in an aqueous environment. Now in this slide, what we're seeing is a representative sample of some simple organic compounds, and this just provides us a sense of the types of structural diversity that we can see in even very small, simple molecules that are based on carbon. What we can see are carbon skeletons of varying lengths, some short, some pretty long, as we see with this fatty acid, palmitic acid. Sometimes we can see just straight linear chains, but other times we'll see branching of carbon chains off of that main carbon chain. And the carbon backbone can even fold back on itself to generate a closed ring structure, like we see here. Carbon can form single bonds and double or even triple bonds, which will also lead to structural diversity, as we'll see in a moment. Now these molecules on the left are all hydrocarbons. You'll notice that they only contain carbon and hydrogen. But if we start including the other 2 common atomic components in biomolecules, oxygen and nitrogen, and thinking about their valence and how they interact to form covalent bonds, we can really start to see how incredibly diverse organic compounds can be. The organic compounds we're seeing in this slide are all small molecules, but as we'll see going forward, some biomolecules are huge and are referred to as macromolecules. This molecule is glycine. It's one of 20 amino acids that are used to build cellular proteins. Proteins are the workhorses of the cell that carry out the majority of cellular functions. We'll look at those in detail not too long from now. Now, an average-sized protein consists of about 300 amino acids linked together end to end to form a chain. So, in terms of size, we're talking about thousands of carbon and hydrogen atoms and hundreds of oxygen and nitrogen atoms in an average-sized proteins. But some proteins are much larger than that, much larger than 300 amino acids. And if we think about the size of our largest DNA molecule, DNA being that molecule in which we store our hereditary information, the instruction manual for the cell, our largest DNA molecule in human cells contains not thousands, but hundreds of millions of carbon atoms. So we're talking big molecules, really big. Now when we're looking at these structural formulas like we see here, it makes the covalent connectivity between atoms very easy to see. But these structures don't really give us an accurate picture of the 3-dimensional structure of real molecules. Again, the functions that these molecules are carrying out in the cell are dictated by their structures and chemical properties. So in order to understand biological function, we must understand molecular structure. To get a better understanding of molecular structure, we need to think a bit more about the arrangements of electron orbitals in covalent compounds. I'm sure you remember that each electron shell beyond the first shell contains two or more subshells with orbitals, or defined regions of space where electrons are most likely to be found. The first shell contains only one spherical 1s orbital, but the second shell contains one spherical 2s orbital and three sort of dumbbell-shaped 2p orbitals. The third shell contains three subshells, and so on. Now, if we revisit the electron configuration of a carbon atom, we'd see, as we said before, 2 electrons in the first shell, and in the valence shell we'd expect to see 2 electrons in the 2s orbital, and then 2 unpaired electrons, each occupying a p orbital. But what happens when carbon forms covalent bonds with other atoms is that these atomic orbitals change shape and morph into hybrid molecular orbitals that depend in number and shape on the number of covalent bonds that are formed between carbon and its atomic partners. As an example, let's look at methane, or CH4, which is carbon singly bonded to 4 hydrogen atoms. The 4 atomic orbitals of carbon's second shell in this molecule actually combine to form 4 molecular orbitals, which are referred to as sp3 orbitals. And they have this characteristic sort of asymmetrical dumbbell shape. Each of carbon's 4 shared electrons is occupying one sp3 orbital in the molecule. And each sp3 orbital is going to overlap the single atomic orbital of each bonded hydrogen atom. And so what you get as a result of those hybrid molecular orbitals is a 3-dimensional shape in which the 4 hydrogen atoms are situated as far apart from one another as possible, so directed towards the 4 points of a tetrahedron in this case, and carbon is at the center of that tetrahedron. As you learned in general chemistry, this is based on electron repulsion. This configuration maximizes the distance between the electron pairs, minimizes repulsion, and therefore is more energetically stable. We can represent this 3-dimensional shape using perspective drawing, as shown here at the right. By convention, these lines represent orbitals or bonds in the plane of the page. This solid wedge represents a bond coming out of the page towards you. And the dashed wedge, or dashed line, represents a bond directed into the page away from you. This type of drawing is really useful for communicating or illustrating 3-dimensional structure in 2 dimensions. Now, what happens if carbon is bonded to fewer than 4 atomic partners? Well, then we see different hybrid orbitals forming. So, for example, if carbon is only bonded to 3 atomic partners, as we see with each carbon in ethane, for example, the 2s orbital and 2 of the 2p orbitals in carbon's second shell will then hybridize to form 3 sp2 orbitals that all lie in the same plane, as shown here. And this is going to yield a molecule that's not tetrahedral, but has an overall trigonal planar 3-dimensional shape. So it's a flat plane, and all of the atoms lie within that same plane. And you'll remember from general chemistry that that third p orbital doesn't disappear, it's unhybridized. So we see a different shape if carbon only has 3 molecular partners, right? Well, what if carbon only has 2 bonded partners? Well, here we see the formation of 2 sp orbitals from the 2s and 1 of the 2p orbitals, and what we end up with is a linear molecule. And you'll remember from general chemistry that the bond angles for these hybrid molecular orbitals are 109.5 degrees, 120 degrees, 180 degrees. We're not going to concern ourselves so much with memorizing those sorts of details. The main point that we want to understand here is that carbon compounds have specific 3-dimensional structures, and those are going to be really important when we start thinking about molecular interactions and reactions that are taking place. Here we're just looking at the geometry around carbon, but if we're thinking about, for example, nitrogen or oxygen, often we're going to see lone pairs that are present. And so these non-bonding lone pairs, as you'll recall, will introduce distortions into these kinds of hybrid orbital structures because of the greater repulsive effects that those lone pairs have. on the other electrons that are being shared. I don't want to belabor this business of hybrid orbitals too much. I don't expect you to memorize bond angles. That's, something that you learned in general chemistry, and you'll look into this in a lot more detail if and when you take organic chemistry. But minimally, what you should recognize here is that when carbon has 4 bonded partners, what we're going to see is a tetrahedral molecular geometry around that carbon. If carbon has 3 partners, that's going to result in a trigonal planar configuration. And if carbon has 2 bonded partners, what we get is a linear arrangement of carbon with those 2 partners. These hybrid orbital configurations were predicted nearly 100 years ago, but only recently it's actually become technologically feasible to visualize molecular structures like these directly, amazingly enough. A 2013 article in Science from Felix Fischer's lab used a technique called non-contact atomic force microscopy to measure the infinitesimally small deflections caused by interactions between a single oxygen atom probe and the atomic components of the molecule that was under study as the probe is moved over the molecular surface. Those tiny little deflections are then used to create an image of the atomic structure of the molecule. Now, interestingly enough, the authors weren't really trying to obtain high-resolution images of actual molecules. What they were just trying to do is to find a way that they could monitor the course of a chemical reaction that they were trying to drive in a particular direction to generate particular graphene molecules. Right? And so they turned to this technique to try and distinguish between different possible products of the reaction that was taking place. But what they ended up generating using this technique is images that were showing for the first time incredibly high resolution molecular structures. And what these images confirmed were those predicted molecular geometries and bond angles to an astonishing degree, really. So what we end up with are molecules that basically look just like the textbook illustrations of those molecules that you'd see in an organic chemistry text. And I just want to draw your attention to this scale bar in these images. That's 3 angstroms, right? That little line represents 3 angstroms. An angstrom, I'm sure you'll remember, is 1/10,000,000th of a meter. So what we're looking at here is visualizations of molecules at the level of 1/10,000,000,000th of a meter. That's amazing. If you're interested in learning more about this technique, just Google the name of it. You can read more about how they use a single oxygen atom as a probe and how that leads to this kind of resolution. It's pretty amazing. All right, so what we can do is we can represent molecules in a variety of ways to communicate whatever specific information we're trying to get across in, some particular context. We can use a molecular formula, like we see here on the left, that shows basically the number and types of atoms that a molecule contains. The structural formula makes very clear the covalent connectivity of the bonded atoms within the molecule. And we can even use a condensed structural formula like we have here on the right that's kind of a combination of those two. It's essentially the molecular formula that also makes those covalent connections a bit clearer. So in this example, we can see that the carbon atom is actually bonded to 3 hydrogen atoms. By placing the hydrogens closer to the carbon, we can see that they sort of belong to that carbon. And then the other hydrogen is bonded to the oxygen. Right, so it just makes it a little bit clearer. If we want to get a sense of 3-dimensional structure, we can use perspective drawing, as we just talked about. And we can also see the use of these ball and stick or space filling models, which are all showing basically the same thing. The ball and stick model, of course, is showing the bonds between atoms a bit more clearly, like the structural formula, than the space filling model does. The space filling model is sort of giving us an overall 3-dimensional configuration. Later in the course, we're going to see some other ways to visualize molecular structures besides these, but these are very commonly used, and you should be comfortable interpreting what you're seeing when you run across these. Now, in the case of this molecule, which is methanol, by the way, given the molecular formula, you automatically know the covalent connectivity and structure, so we wouldn't actually ever need to use a condensed structural formula. Why? Well, because there's only one way that we can connect 1 carbon, 4 hydrogens, and 1 oxygen, such that each of those atoms' valence is satisfied. All right? But what if I gave you the molecular formula of a larger molecule? Let's say C4H10. would you necessarily know its structure? Well, not precisely, because if you think about it, there's more than one way that those atoms could be arranged. When molecules have the same number and types of atoms, but a different covalent connectivity or a different arrangement in three-dimensional space, we call these molecules isomers. So we're going to talk about a couple of different types of isomers that you should be familiar with. We're going to start here with what are called structural isomers. So here we have two different versions of molecules that are both C4H10. And what you can see is that they have a different covalent connectivity of those atoms with one another. The structure on the bottom is a single 4-carbon chain, while the structure on the top is a 3-carbon chain with another carbon branching off of that central carbon. So these types of isomers that have a different covalent connectivity between the atoms are structural isomers. Now looking at these, would you predict that these molecules would have the same chemical and physical properties or not? Well, they're both hydrocarbons. They're both made up of only carbon and hydrogen, so we could probably think about, their essential hydrophobic nature as being the same. But let's think about this branching. If you consider that, you probably predicted that the introduction of branching in this molecule is going to disrupt intermolecular interactions, right? Whereas this linear chain is going to be able to more effectively stack one molecule on top of the other. So if we're thinking about physical properties like boiling point, for example, we'd expect them to be different because we're going to see different intermolecular interactions that are possible based on their different structures. So same molecular formula, different physical structure, and different chemical and physical properties potentially as a result. Now, sometimes you can have isomers with the same covalent connectivity, but different spatial arrangements of the atoms, and these are called stereoisomers. In other words, if you look at each atom and what it's bonded to, there's no difference, but the atomic partners are arranged differently in three-dimensional space. There are two types of stereoisomers that you should be aware of for 204. The first are called cis-trans isomers. These become possible when molecules have rotational restriction around one or more bonds, which we see when there are double or triple bonds or ring structures in a molecule. Without going into too much gory detail, this is a result of sp2 and sp hybridization of molecular orbitals, and more specifically the fact that when we have that kind of hybridization, there are unhybridized p orbitals which share electrons in side-to-side orientation rather than head-to-tail. And this is in contrast to single-bonded atoms, which do have free rotation around the bond. If you haven't had OChem and you didn't learn much about molecular orbitals and general chemistry, don't worry too much about this. Suffice it to say that when we're looking at organic compounds where double or triple bonds or ring structures are present, there will be rotational restriction. So here, for example, in this dibromobutene molecule, there are two possible isomers that exist. One in which the two bromine atoms are positioned on the same side of the molecule relative to the double bond, as we see here. This is called the cis orientation, cis meaning same. And the other one in which the two bromine atoms are situated on opposite sides of the molecule relative to the double bond, in the trans orientation, trans meaning across or opposite. Just like we saw in the example of structural isomers, this difference in orientation, cis versus trans, has important impacts. It may influence how these molecules interact, as well as whether a dipole moment cancels due to symmetry or not. And so these may result in different properties like density, melting point, boiling point, etc. So if we're naming these molecules, it wouldn't be sufficient just to say dibromobutene. We'd have to specify whether we're talking about cis dibromobutene or trans dibromobutene, because spatial orientation matters. Enantiomers are a type of stereoisomer in which we have the same atoms with the same structural connectivity, but that result in mirror image isomers that can't be superimposed on one another. And this usually comes about when there's what's called an asymmetric or chiral carbon, which is a carbon that is bonded to 4 different atomic groups. So again, in order to be chiral, a carbon atom must have 4 bonded partners. You kind of have to imagine yourself as the carbon atom and look down each of the bonds that it has and see if you see something different at the end of that bond, taking into account all of the atoms at the end of that bond. When that's the case, the carbon is chiral, and the possibility of mirror image isomers or enantiomers is introduced. The classic example of non-superimposable mirror images is your right and your left hand. There's no way that you can turn or rotate your hands in such a way that they are oriented the same way in space and superimpose on one another. Enantiomers are the same exact idea at the molecular level. Here's an example. So we see a chiral carbon here bonded to 4 different groups of atoms, a hydrogen atom here, a carboxyl functional group here, an amino group here. We'll talk more about functional groups in a bit. And then another group of atoms that's not specified but is symbolized by the letter R, which is different from the other 3. The molecular geometry around this carbon is going to be tetrahedral, with these 4 bonded atoms or groups of atoms oriented in 4 different spatial directions. Now we have to imagine if we had a mirror, this molecule on the left would be what we would see in the mirror, right? That's what we mean by mirror image isomer. There's no way to rotate this version of the molecule on the right so that it's superimposed and spatially arranged in exactly the same way as the one on the left. Now, interestingly, enantiomers have identical chemical and physical properties in all but one way, and that's with regards to their interactions with plane polarized light. It turns out that when a chiral center exists and is exposed to plane polarized light, one enantiomer will rotate the light to one direction to some degree, and the other enantiomer will rotate the light to exactly the same degree, but in the opposite direction. And the naming conventions that were initially used for enantiomers reflected that property. Enantiomers were referred to as the plus and minus isomers, or L and D, which stands for levo and dextro from the Latin meaning left and right. In organic chemistry, chiral centers in organic compounds will be assigned as R or S based on atomic priority of the bonded atoms. But in biology, enantiomers are commonly referred to as the L and D isomers, both capitalized. And this is actually based on comparing this structure to a reference molecule called glyceraldehyde. Again, these are details that are not so important as recognizing the important concept here, which is that whenever a molecule has a chiral center, the possibility of mirror image isomers exists. This business of mirror images in 3-dimensional space is a bit tricky to visualize in 2 dimensions. So if you find this confusing, don't worry. We're going to do some model building in class to really solidify your understanding of this concept. Now, if enantiomers have the same physical and chemical properties, except for rotation of polarized light, why are we spending so much of our time talking about them? Well, because biologically speaking, enantiomers are completely different molecules and cannot substitute for one another in terms of their molecular interactions and reactions. In fact, each enantiomer is biologically distinct and would be expected to have distinct interactions in a living system. In other words, biological interactions are extremely stereospecific. A simple example that illustrates this point is shown here. Let's say that we have a particular compound that acts as a signaling molecule, and it binds to a cellular receptor, and when it binds, it causes the cell to respond in a particular way. Now let's imagine that this central sphere is the carbon atom, and each of these shapes represents a particular set of bonded atoms connected to that carbon. Notice that the binding sites shown on the receptor down here, which are simplistically represented as a square, triangle, and a circle, well, the matching parts of the signaling molecule are going to align with these binding sites and are going to be able to dock onto the receptor in this highly specific manner to initiate the response. But look at this mirror image isomer or enantiomer of the signaling molecule shown at the right. Although it has the same groups, it has a different arrangement of those groups around that central carbon. And so the groups don't match up correctly, and there's no way that we can rotate this molecule to make it match up. So the enantiomer on the right would not be able to activate this receptor. However, it's important to note that there may be some other receptor or molecule that the enantiomer on the right would be able to bind to, but the isomer on the left wouldn't. Again, biological interactions are highly stereospecific. This has relevance not just for understanding biological interactions in general, but also from a clinical perspective. So let's say you're a researcher that's working for a pharmaceutical company and you're trying to synthesize the compound on the left to specifically affect a particular cellular response. The fact that there's a chiral center means that enantiomers will be present, and in the best case scenario, the wrong isomer has no effect on its own, but will reduce your yield of the enantiomer you're trying to synthesize. So synthesis methods have to be stereospecific themselves in order to yield one specific enantiomer, but not the other. In the worst case scenario, however, the wrong enantiomer has biological effects of its own, and these off-target effects can range from mild to very, very bad. An example of this latter scenario happened in the 1950s with the drug thalidomide. Thalidomide is a compound that was developed and marketed in the 1950s in what was then West Germany as an effective sedative and antiemetic, which means that it reduces nausea and vomiting. It was hailed as a wonder drug and was prescribed broadly in 46 different countries, including to pregnant women to reduce the severity of their morning sickness, which it did. Unfortunately, it soon became apparent that the drug crossed the placental barrier and severely disrupted fetal development, particularly if the drug was taken during a specific window in the first 3 months of pregnancy. Many women miscarried. We don't actually know how many, but there were about 10,000 babies that were born worldwide with severe birth defects caused by this exposure to thalidomide. And these included blindness, deafness, various organ malformations, including malformation of the limbs, which we see here in these kids having a swimming lesson. Most of these kids were in West Germany, where thalidomide was actually available over the counter. You didn't even need a prescription. Only about a half of the infants that were born survived. Now, why the extremely devastating effects? What's going on here? Well, it turns out that thalidomide at the molecular level has a chiral center. Here's the structure, and the chiral carbon is here. Don't worry if that isn't immediately apparent to you. It's tricky with ring structures to recognize the chiral centers. Just take my word for it. This version is the sedative, and this version causes birth defects. How it does that is not actually known precisely, but it is known that this enantiomer of thalidomide disrupts growth factor signaling that's critical for normal development. Both enantiomers were present in the prescribed drug, and so that's how the fetuses were exposed. But in this case, it actually wouldn't have mattered if the synthesis had been developed to be specific for only the R enantiomer. In other words, for only the enantiomer that has the sedative effects, because in the body it turns out that this compound will freely interconvert from one isomer to the other. And that's actually not that uncommon. So take-home message, biological interactions are very dependent on structure and spatial arrangements within molecules. If you have different structures or different three-dimensional arrangements, you get different interactions and therefore different biology. Okay, so we've seen that organic compounds are incredibly diverse structurally. It seems that there are an infinite number of ways that molecules could be put together using the basic atomic building blocks of carbon, oxygen, nitrogen, and hydrogen. So how can we ever hope to understand molecular interactions if that's the case? Well, it turns out it's not as bad as all that, because what we see in biomolecules are recurring groups of atoms that will have particular properties and engage in characteristic types of reactions and interactions whenever we see them. And these are called functional groups. So here in the last part of this video, I'm going to introduce you briefly to 7 specific functional groups that we see recurring over and over again in biomolecules. Again, these are not found in isolation. These are groups of atoms that are part of a larger organic molecule that we're just going to call R, but they contribute specific properties to that larger molecule whenever they're present. These functional groups also participate in characteristic types of chemical reactions with one another, but that's an OChem topic, and in this class we'll be focusing not so much on reactions between them, in other words, reactants being converted to products, but rather those intermolecular interactions that we talked about before, things like hydrogen bonding, things like dipole-dipole interactions or dipole charge interactions. Now, I'm only going to give the briefest of brief introductions to these now, because we're going to spend quite a bit of time in class delving into these functional groups and their properties and their interactions more deeply. First up in the list is the hydroxyl group. The hydroxyl group contains an oxygen atom covalently bonded to a hydrogen atom and obviously also connected to the rest of the molecule, again, that we're calling R. Note that this is not a hydroxide ion. This is a functional group that's connected to a larger organic compound. Now, we've already talked about the nature of the covalent bond between oxygen and hydrogen in a water molecule. Based on those discussions, what predictions would you make about the properties of this functional group when it's found in an organic compound? Next up is the carbonyl group, which consists of a carbon atom double bonded to an oxygen atom. Depending on the location of the carbonyl group along a carbon chain, these will either be classified as aldehydes if they're at the end of the carbon chain, or ketones if they're internal to the chain. How do the electronegativities of carbon and oxygen compare? What properties do you predict for this functional group? Now, both hydroxyl and carbonyl groups are found in all sugars, but they're also found in lots of other molecules as well, and we'll see them over and over as we take our tour through biomolecules. Carboxyl groups, or carboxylic acids, consist of a carbon atom double bonded to an oxygen and also singly bonded to the oxygen of a hydroxyl group. Now, at physiological pH, these groups will actually ionize, with the hydroxyl losing a proton. This is why they're referred to as acids, because they increase the hydrogen ion concentration in solution. So at pH 7, like we'd see in most cells, this functional group is ionic and contains a full negative charge. Amino groups consist of a nitrogen atom bonded to two hydrogen atoms. Now, these functional groups will also ionize at physiological pH, but they're basic, and so they tend to pick up a proton from solution, and what we end up with is NH3+, an ionic functional group with a full positive charge. Carboxyl groups and amino groups are components of all amino acids, hence the name amino acid, but they're found in other molecules as well, so we'll see them again. Sulfhydryl groups consist of a sulfur atom bonded to a hydrogen atom. And while these are slightly polar depending on the molecular context, more importantly, they can undergo oxidation reactions to yield disulfide bonds in which the hydrogens are eliminated and the two sulfur atoms are now bonded to one another. We'll talk about these more when we discuss proteins in more detail, because disulfide bonds like these are found in proteins where they stabilize protein folding and therefore the overall structure. Methyl groups consist of a carbon atom bonded to three hydrogen atoms. Now, with only carbon and hydrogen, do you predict that this functional group is going to be polar or nonpolar, hydrophilic or hydrophobic? Would you predict that the presence or absence of a methyl group can influence the properties of a molecule that it's a part of? Finally, phosphate functional groups consist of a phosphorus atom covalently bonded to three oxygen atoms, and these functional groups contain two full negative charges at pH 7. So they, like the carboxyl and amino groups, are ionic. They're negatively charged. Phosphate groups are found in the subunits that make up DNA and are important contributors to the way that the structure forms, and we'll talk about that more later on in the course. There are a couple of other contexts in which the structure and properties of phosphate functional groups become important. Specifically, these are related to the regulation of molecular function and in energetics. So I just want to introduce these very briefly, but again, we'll talk about them in a lot more detail later on in the course. So first of all, reversible covalent addition and removal of phosphate groups is a very commonly used mechanism for regulating protein function in cells. So for example, let's say you have a protein in a cell that carries out some critical process. Let's say it's an enzyme that catalyzes some chemical reaction or speeds up some chemical reaction. But the cell only needs for that to happen under particular conditions. Well, the cell can use other proteins to add a phosphate group to this enzyme, and that's called phosphorylation. And this can be used as a type of a molecular switch to turn the protein's function on or off. And we see this a lot in biology. This is a very, very commonly used regulatory mechanism. The other context in which phosphate is really, really important in a biological context is in energy transfer reactions. At some point in your education, I'm sure you probably heard of a molecule called ATP, which stands for adenosine triphosphate, commonly referred to as the energy currency of cells. Here's the structure of adenosine triphosphate. This is the adenosine part, and here are the three phosphates. For reasons that we'll discuss later, when the bonds between these phosphates are broken, that's an energy-releasing reaction. And cells can link this energy-releasing phosphate cleavage to other reactions and processes that are happening in the cell at the same time to provide the input of energy that's required to make them happen. Most commonly, it's the terminal phosphate that's being cleaved off to yield adenosine diphosphate, or ADP. The cell is constantly recycling the pool of ADP and phosphate to reform ATP. But of course, this must then be an energy-requiring reaction. So where does the energy come from to rebuild ATP? Well, it comes from breaking down other organic compounds, and that process releases energy. So in us, for example, when you take that trip to In-N-Out, that provides you with various organic compounds that contain energy. You break them down in the digestive tract into small molecules, which are then absorbed into cells, which then oxidize them to provide the energy to rebuild cellular ATP from ADP and phosphate. And so this is a cycle. We're constantly recycling this pool and using the energy that's been acquired from the outside environment. So energy transfer is another critical role that we see phosphate functional groups involved in biologically. Well, this has been a whirlwind tour of functional groups, but don't worry, as I said before, this was meant to be a very brief introduction, and we're going to spend a lot more time focusing on these in class. We've discussed a lot of topics in this video, but the main ideas we've covered include the relevance of carbon's atomic structure and properties to the organic compounds it's a part of. We delved into the three-dimensional structures of carbon compounds, and we saw how molecules with the same atomic components could actually be structurally or spatially distinct from one another. And I provided a very brief introduction to the most commonly encountered functional groups in biomolecules. Building on these concepts, next we're going to talk about the structures and properties of the four main classes of biological molecules that cells are made of, beginning with the carbohydrates and lipids.